Hi, welcome to my YouTube channel. Today through this video, I'm going to give you some idea what type of safety measure you have to take in chemical as well as biological laboratory. Before going into the details, let us start with few questions. Number one question is, write the meaning of the following symbol in one word. There are four symbols A, B, C as well as D. Followed by next question is on how will you minimize the risk of hazard in the laboratory. Then the next question is write the purpose on use of the following globes in a laboratory. There are two types of globes, A as well as B. After watching this video, you will have the idea on what are the safety guidelines, then categories of the lab hazards, then material safety data sheets, how to store some materials, especially chemicals in a particular laboratory, what type of safety precautions you have to take, then personal protection equipment, how to minimize lab bio as well as chemical hazards, followed by keys to working safely with biohazards. Let us start with the purpose of the safety guideline. Number one is to promote self-awareness and encourage safe working practices in a particular laboratory. Followed by, as a reminder that can be done to work more safely and are applicable to all users of the laboratory. Followed by another purpose, the research worker should adhere to safety guidelines and maintain safety standard very strictly while working in a laboratory. It may be biological laboratory, it may be chemical laboratory. There are mainly three categories of laboratory hazards. Number one is biological hazards, followed by chemical hazards, and followed by physical hazards. And for all these three hazards, this type of symbols are used. So we have to remember the meaning of these symbols. Number one is biological hazard, followed by chemical hazard, and followed by physical hazard. Chemical hazards includes a wide range of chemicals that are used in research laboratory, each with its own inherent hazards. An understanding of the potential hazards and the precautions required in handling of chemical is important in preventing exposure to the chemical and mishaps. There are different types of chemical hazards. Number one is reactive, number two is flammable, number three is toxin, and followed by corrosives. There are different routes for chemical exposure. Number one is inhalation directly into the lungs, as shown in the pictures, followed by absorption through skin membrane or cut into the skin. It is called your direct contact. Then followed by ingestion. It is mainly via mouth into the gastrointestinal system. Corrosives are the most common chemical hazards. These are the substances that cause a destructive burn on the tissues by chemical action at the site of contact. Corrosive effect can also occur in the respiratory tract in case of inhalation and in the gastrointestinal tract in case of ingestion. Sulfuric acid, nitric acid, potassium hydroxide, and sodium hydroxides are common examples of corrosives. Followed by oxidizers or the reactives. These are the chemicals, especially the oxidizers are the chemicals that decompose under certain conditions to yield oxygen. They can cause a fire to burn violently. Oxidizers must not be stored with flammables. Hydrogen peroxide, nitric acid, perchloric acid, nitrates, etc. are the common example of oxidizers. Then what do you mean by MSDS, that is Material Safety Data Sheets? MSDS is the informational document prepared by the manufacturer or the importer of a hazardous chemical, which describes its physical and chemical hazards and recommended precautions for storage, handling, as well as disposal. The main purpose of MSDS is to provide information on healthy and physical hazards. And where are they should kept in a particular laboratory? These are actually kept in notebook, in prep room, or these are also available in online. So always we have to ensure that MSDS is obtained with the chemical and is readily available for reference by the laboratory users. MSDS generally have different type of symbol, 1, 2, 3, as well as W, and which is crossed. 1 means reactivity. So these have number of grades 0 to 4. 4 means may detonate. 3 is shock and heat may detonate. Then 2 means violent chemical changes. 1 means unstable if heated, and 0 means stable. Followed by 2, which is marked with red color, that is included for fire hazards. So there are different flash points from 0 to 4 for fire hazards material. 4 means below 73 degree Fahrenheit. 3 means below 100 degree Fahrenheit. 2 means below 200 degree Fahrenheit. And 1 means above 200 degree Fahrenheit. And 0 means will not burn. Followed by 3, which is marked with blue color. These are health hazards and marked from 0 to 4. 4 means deadly, 3 means extreme danger, 2 means hazardous, 1 means slightly hazardous, and 0 means normal material. And in MSDS, another symbol is generally used, W with cross or strike through. So these are called specific hazard. OX means oxidizer, W with strike through, use no water, and SA means simple asphyxiant, means a substance or the mixtures that displaces oxygen in the ambient atmospheres. These are called simple asphyxiant and can thus cause oxygen deprivation in those who are exposed, leading to unconsciousness as well as death. So this type of material should be always marked very nicely. So now question arises, how to store the chemical in a particular laboratory? So always we have to store the chemicals separately, which are compatible group, and incompatible group should be stored separately in separate cabinet or the shelves. So always we have to maintain the alphabetical order within the compatible group while storing the chemical in a cabinet or the shelves. The number of the example of the chemical group which must be separated by storing them in different cabinet or the shelves or by providing secondary containment. Number 1 is oxidizer, including peroxidase, followed by acids, base, flammable material, reproductive toxins, then carcinogens. So any incompatibles within the above group must be stored separately in separate cabinet or the shelves. Chemicals must not be exposed to heat or the direct sunlight. Flammable solvent must not be left open in containers. And after use of each and every chemical, they must be immediately removed to the safety. There are a number of safety precautions that should be remembered by a laboratory worker while working in a laboratory. Number 1 is do not work alone in the laboratories, particularly when performing hazardous procedures. You should plan appropriate procedures and positioning of all equipment before beginning of any experiment. Always wear appropriate personal protective equipment at all the time in the laboratory. You should know the locations of emergency equipment. Always be aware of the hazards posed by the work of others in the laboratory. Please keep your work area clean and uncluttered. And always that laboratory equipment must be regularly inspected and serviced as per the manufacturer recommendations. Please keep coats, bags, and other personal items in the designated area, but not on the bench top. Please try to remember, safety is part of science. Now, let us try to understand something about personal protective equipment. It includes number one, that is called your protective clothing, that is called your lab coats. So it creates a barrier between us and the experiment, and by wearing lab coat we can prevent our body from many accidents. Number two is sleeves. These are recommended for reduced skin exposure to chemicals. Next is aprons. It protects from the chemical splashes. Next is shoes. It may be needed if leaving the environment that is infectious or entering a sterile environment. And for better understanding, please see the photograph. So here, while working in a laboratory, for girls or the persons who has long hair, they have to tie it back. Don't forget to wear safety glasses, lab coat, gloves, long pant, and closed-toe shoes. Another personal protection equipment in the laboratory is eye and face protection. It includes goggles, and these goggles should be always appropriate for the task you are doing. It may be your safety glasses, splash glasses, and there are some face shield. And these goggles, face shield, then safety glasses or the splash glasses protect our eyes and faces from the hazardous material. So now, how we can protect our hands? So gloves is the ultimate, and gloves will provide the protection against corrosive or toxic chemicals, biological contaminants, sharps, extreme temperatures. There are different type of gloves. Number one, thin wall gloves that still provide dexterity. Then thermal gloves for high temperature. Then cryogenic gloves, rubber gloves for dishwashing. Then puncture resistant gloves, especially for handling animals. So these are the different type of the gloves that are used in different type of laboratory. Then mask and respirators. So there are different type of mask. Let us try to understand what are those. Number one is surgical type of mask. Here the filter that are present in the mask, okay, it prevents the dust and the aerosols. Then shield face, it protects us from the minor splashes. Then little protection from the infectious agent. Followed by respirator. It is a type of breathing devices and it's designed to reduce airborne hazards by manipulating the quality of airs. So while using respirator by a laboratory personnel, they must be well trained. Otherwise, there may be a breathing problems. Now, let us understand how to minimize the hazard in a laboratory. So for that, some suggestion I have included. So please try to listen. Number one is your work area should be prepared very nicely before you start off your work. Always try to use chemical fume hood, while handling hazardous chemicals. Try to limit the exposure to hazardous material. You should store the chemical properly in designated shelves and cabinets in alphabetical orders. Try to handle the waste properly, and they should be thrown in designated bins. We should have proper knowledge on hazardous material and non-hazardous material that are generally used in a particular laboratory. We should not have food or the drink anything in the laboratory. We should follow the instruction of a laboratory or the guideline of the laboratory very carefully as well as strictly. You should always read the labels that are generally pasted in a particular laboratory. You should never taste, smell anything in a laboratory. Always you should wash your hand after each and every experiment. Please try to read the lab before performing or paying special attention to the safety sections. Please try to place books, backpacks on tables away from the lab benches during the experiment. Now let us understand an emergency what we have to do. Suppose our body, eyes or the skins it has been exposed to some chemicals. Immediately what we have to do. So these are called emergency measures. Number 1, all chemical splashes on the skin must be immediately flushed under running tap water, as I have shown in the pictures. Next, contaminated clothing must be removed while flushing the body, and flushing must be continued for at least 15 minutes to remove all the traces of the chemicals that came in contact with your eyes or the body skins. Eye must be immediately flushed with water for at least 15 minutes. And next, always try to refer safety data sheet for more and more information. Now, another important point we have to remember while working in a laboratory, that is called your disposal of the chemicals, which is important part. So in a particular laboratory, they must maintain labeled carboys or the cans for collecting spent chemicals. Care must be taken to prevent mixing of incompatible chemicals while transporting the spent chemicals. Otherwise, some accident may happen. There should be at least 2 inch head space above the liquid surface in the chemical container. In spite of following all the guidelines or the safety measures, a particular lab worker may get exposed to chemical hazards. So in that case, how we have to respond to that chemical hazards for a particular laboratory, for a laboratory workers? So first of all, our duty is to notify the instructor of the particular problem and make others in the vicinity aware about the incident. Then we have to understand how to handle the chemical spills. We should have proper knowledge or the information or the awareness about the assess the situation, and we have to evacuate as soon as possible if necessary. We have to remove the cloth. That should be done very correctly, and there must be a chemical spill kit in the laboratory so that during urgency that can be used immediately by the lab worker. Let us discuss about biohazards. These are the infectious agent or the other biological material that present a risk or potential risk to the health of the human, animal or the environment. It includes the organisms and the viruses infectious to the human, animal or the plant, cultured human as well as animal cell. So these organisms includes parasites, viruses, bacteria, fungi, prion and rickettsias. Certain type of recombinant and or synthetic DNA are also included in the biohazardous material. Then biologically active agent that may cause disease in other living organism or that cause significant impact to the environment or the community. The common example are toxin, allergens, venoms and many more. Biohazards have number of risk groups. These risk groups are designated as 1, 2, 3, 4 based on potentiality to cause disease to human, hazard degree to worker, spread to the community, and effective treatment or the preventive measures. Say, let us discuss with the risk group 1. For first category, it is unlikely potential to cause disease to the human, followed by low hazard degree to worker, then unlikely spread to the community, and then effective treatment for preventive measures. These are available. But in case of 4, here the potentiality is that likely to cause serious diseases, but treatment or the measures may not be present. That's why it is marked with the red color 4, and the risk group is 4. Then in case of hazard degree to the worker, it is very serious, spread to the community, likely to be spread, and then effective treatment or the preventive measure, it is unavailable. That's why it is marked with red. But in case of 3, it is a little bit less dangerous as compared to the 4, and then followed by 2, and then 1 is a little bit safely. There are 4 categories of biosafety levels. Biosafety level 1 to biosafety level 4. Biosafety level 1 is for low risk microbes. On the other hand, BSL4, that is biosafety level 4, is for high risk microbes. And BSL1 is not associated with the disease in healthy adult. In case of BSL1, Escherichia coli, yeast, and most plants are included, and these are handled in BSL1. And BSL2, these are associated with the disease that is treatable or vaccine are available. The common example is Salmonella and Clostridium. And in BSL3 and BSL4, here we used to handle the severe lethal human diseases. It includes Mycobacterium tuberculosis, Ebola, and many more. So these type of symbols are generally used in different type of biosafety level. BSL1 green color, BSL2 light yellow color, BSL3 reddish color, and BSL4 red colors. So BSL1, already I said that these are low risk to personnel and the environment. And BSL2, these are moderate risk to personnel and the environment. BSL3, these are serious disease for human, animal, or plant and not spread by causal contact. On the other hand, BSL4, these are very serious disease for human, animal, or the plant, and often untreatable. So this type of organisms are generally handled in BSL4. So how to minimize the risk of biohazard in a particular laboratory? Before going into the details, please see the diagram. From the diagram, you could be able to understand how we can minimize the risks of biohazard in a particular laboratory. Still, we have to follow some of the important points to minimize the risk of biohazard. Number 1, always follow the standard microbiological practices while working in biolaboratory. The biolaboratory should have limited access. All workers should wear personal protective equipment while working in the laboratory. Eating and drinking should not be allowed in a particular laboratory. Hand to mouth and hand to eye must be avoided in a biolaboratory. You should not follow the mouth pipetting. Try to minimize the aerosol. Try to disinfect work surface before and after lab, and try to decontaminate all live material, especially DNA, RNA, before its disposal. So these are the points we have to remember to minimize the risk of biohazards in the laboratory. Besides that, to minimize the risk of biohazard in the laboratory, the design of the laboratory should be appropriate. Try to use biological safety cabinet based on the need. There are 3 different type of biosafety cabinet: class 1, class 2, and class 3. As per the need of the laboratory worker, this type of facility must be there in the laboratory. You have to prevent the bioaerosol production. Use of disinfectant and sterilization is must, like bleach, ethanol. Carefully handle the biohazards waste, which is very much important for a biological laboratory. And these are some of the example of safety equipment which must be there in a particular laboratory. Number 1, facility for eye wash, facility for safety shower, fume hood, fire extinguishers, biosafety cabinet, short containers, fire blanket, and many more. So a number of keys are there while working safely with biohazardous material. Number 1 is performing a risk assessment prior to beginning work with biohazardous material or when procedures and protocol have changed for an established SOPs. That is standard of procedure. Secondly, understanding what risk group and biosafety levels are and the differences between them. Next is ensuring that training requirements have been made prior to beginning work with biohazardous material, followed by ensuring that any required equipment has been certified and is in proper working order, followed by ensuring that any required PPE is available and that laboratory personnel are trained in its proper use and care. Then posting biosafety signs in appropriate location throughout the lab and especially at any entrance to the lab. Besides that, the principal investigators and the lab group need to take the following action. Number 1, register your lab in the laboratory registration system. Establish and maintain laboratory specific risk assessment policies and procedures and update them as necessary to optimize effectiveness. Followed by ensure that all laboratory personnel have completed the required training prior to beginning work in the laboratory. Reinforce any new annual or refresher training that may be necessary. And they must conduct an inventory of all infectious agents and toxins. They must properly dispose of all legacy culture and samples, maintain a record of all the infectious agent and update information in the laboratory registration system. With this, I would like to thank you all for watching this video. Thank you.